Okay, so I think we can make a start. So, good afternoon, everyone, and welcome to the Palaeoverse Lecture Series. So great to see so many of you here today. So, this lecture series is focused on software development and methodological approaches in paleontology, with the aim of promoting the use of new tools in the field and to give researchers a platform to showcase their work. With this series, we hope to shake up the usual type of lecture by having a mixed format with time dedicated to the presentation, demonstration, and discussion of the software or method presented. Now, typically these sessions will run for about 50 minutes in total. I will run this series throughout the year, generally on the last Thursday of each month, unless they overlap with holidays. And if you're interested in presenting a new method or software you have been developing or implementing, please feel free to reach out, as we're always happy to hear from the community. So before moving on, just a few quick introductions. My name is Lewis Jones, and with me today I have Erin Dillon, Bethany Allen, Harry Drage, and William Gertie. The five of us are on the core team of Palaeoverse, an organization aiming to bring the paleobiology community together to share resources, reach agreed standards, and improve reproducibility in paleobiological research. And if you want to know more about Palaeoverse, please check out our website, which I believe the link is being posted in chat right now, and our GitHub organization. So that's all from me today. Harry will now give you some helpful tips and instructions for this webinar, and Erin will introduce this month's speaker. Thank you very much, and please enjoy the lecture. So over to you, Harry.
Hey, everyone. So as Lewis mentioned, the format of this seminar consists of a talk by our speaker, followed by 10 to 15 minutes at the end dedicated to a Q&A and discussion. And my fellow hosts and I will help field your questions and moderate this discussion. Please note that the seminar will be recorded and be made available on the Palaeoverse YouTube channel. However, we'll stop the recording before the Q&A period in case you'd prefer not to be recorded. But before we get started, I'd like to quickly point out a couple of useful controls at the bottom of your screen. There is a Q&A button, so you can use this at any time to submit questions to our speaker. And during the Q&A session at the end, we'll either read out your question or invite you to turn on your microphone if you'd like to ask the question directly. There's also a raise hand button, so alternatively during the Q&A you can raise your hand and we'll call on you to turn on your mic so you can ask your question yourself. There's also a chat, and currently you can only chat with the hosts in case you have technical difficulties, but during the Q&A period we'll allow you to chat with other attendees as well. And there's also a button for closed captions, so you can turn that on and view the transcript at any time. Lastly, I'd like to highlight a couple of ground rules. Our aim is to cultivate an open, respectful, and inclusive environment during this lecture series where we can all learn from each other. So with this in mind, we ask that you please be respectful and kind when engaging with the speaker and with fellow attendees, particularly when you're asking questions and interacting with others in the chat. Any form of harassment or inappropriate behaviour will be grounds for removal from the webinar platform. And for more information, you can refer to the Palaeoverse Code of Conduct, which is on our website, and we'll also put a link in the chat now. And with that, I'll pass you over to Erin, who will introduce our day's speaker.
Awesome. Thanks so much, Lewis and Harry, for getting us all up to speed on how the lecture series works. So today I have the great honor of introducing our speaker, Amy Shipley, who is a PhD student at the University of Leeds, working with Dr. Alex Dunhill. So prior to moving to Leeds, Amy did her undergraduate work in zoology at the University of Reading and obtained a master's degree in paleobiology from the University of Bristol. And we just learned this morning that she's in the final stages of finishing her PhD, which is very exciting. So Amy is doing some really spectacular work modeling ancient ocean food webs to examine the trophic consequences of extinction. In particular, her dissertation work has focused on the Pliocene marine megafaunal extinction event, during which one of the largest apex predators of all time, the megalodon, went extinct. And I think we'll be hearing about some of this work today. So without further ado, we're really excited to hear from Amy with a talk titled Modeling Ancient Food Webs, Ecosystem Changes Across the Pliocene Marine Megafaunal Extinction. Please join me in welcoming Amy to our virtual stage.
Great, thank you so much. I'll just quickly share my screen. Hopefully everyone can see that. So, thank you so much for having me. So today I'm going to be talking about one model that you can use to reconstruct paleo food webs and how this fits into the larger remit of my research on ecosystem changes following a Pliocene marine megafaunal extinction. So before I start, I just want to quickly acknowledge everyone that's been working on this project so far with me. So my research is largely interested in the impacts of marine megafaunal declines on marine ecosystem structure. So currently, marine megafauna are undergoing really significant population declines, and this is happening globally, and it's due to a lot of different factors. Just to name a couple now, this has been due to overfishing. So not only is it as a response to loss of prey populations, but also when marine megafauna are accidentally caught as bycatch. And then also we have climate change and population causing a lot of habitat degradation and reducing range habitats. So currently there's around 1/3 of all marine megafauna today that are at risk of extinction, but we don't really understand how this will impact marine ecosystems if they do actually go extinct. So my larger research question is, how do marine megafaunal extinctions impact marine community structure? And it is really important to consider marine communities when we're looking at biodiversity loss, because even if you are just seeing extinctions in one small group, so in this case marine megafauna, there will be cascading impacts which do have the potential to have much wider reaching effects on ecosystems, potentially impacting their structure, function, and even how stable they are and robust to future losses. So when we look at community structure in the form of food webs, so food webs just show the feeding interactions between different taxa in the community. Megafauna normally sits within the middle or the higher trophic levels. So another way to phrase this question would be, what happens to food web structure if we lose a bunch of these nodes in the mid and high trophic levels? So to address this question, I've been looking at a Pliocene marine megafaunal extinction event, and this happens at the very end of the Pliocene, so around 2.6 million years ago. And it's really interesting because as far as we can tell, this doesn't really seem to impact any other group. We only see losses in marine megafauna, with around 36% of all marine megafaunal genera going extinct globally. And this is quite unusual for extinction events. And the proportion of megafauna loss that we do see is very similar to the proportion that are currently at risk of extinction. So it makes it a really great analog. So during its extinction, it was predominantly cetaceans that went extinct, but it also included the giant shark Catodus megalodon, which was the apex predator at the time. So to address this question, I have been reconstructing paleo food webs. So paleo food webs can be really great at looking at ecosystems before and after an extinction event, because it kind of gives us this snapshot in time of what this community looks like before and after, and then we can quantitatively measure the changes in ecosystem structure and function. And there's a whole bunch of different ways that you can model food webs. I know Roxanne Banker gave a really great talk at the end of last year talking about how they reconstruct paleo food webs in their research. But today I'm just going to be focusing on the model which I use, which is the paleo food web inference model. So this is a relatively new model, and it's trait-based and specifically designed for fossil data. And the way it works is that it assigns feeding links based on rules which are grounded in optimal foraging theory. So the FIM, the paleo food web inference model, generates structural food webs. So what I mean by this is that these are topological webs which only consider the presence or absence of taxa. So there isn't a measure of biomass or population data. Because of this, when I show you my webs, we have one node at the very bottom which routes the web, which just represents all primary productivity going into this web. But the amount of biomass or productivity isn't quantified. And using the FIM, we're able to generate two different types of food webs, and I'm going to go through both of them today. So the first one is a meta-community food web. So these are food webs which show all possible feeding interactions within a community. And then the second is realized food webs, which are essentially these meta-community food webs which have been downsampled to reduce the link distribution. So starting off with the meta-community food webs, the FIM model requires two different bits of data. So firstly, it needs a taxonomic list with ecological trait data. So it could look something like this, for example, in the top right, where you have your clade, and then there are 4 different ecological traits. And then the second bit of data that the FIM needs is a list of rules for each of these traits, which determines whether or not a feeding interaction is feasible. So for example, a very common trait is the trait of body size, and the rule that's often used with body size is that a consumer can only feed on a resource if the resource is the same size as it or smaller, because this is the way that it's only energetically feasible. So once you have this data, all of the modeling is performed in R. And what the model does first is it creates pairwise interactions between all of the taxa in the community. And it will then go through each of the different traits to determine whether or not a feeding link is feasible. So for example, if your trait is body size and your rule is what I just said, so a consumer can only feed on a resource if it's the same size or smaller than itself. In this case, only the top 2 consumers can feed on this resource. So the model then does this for every trait, and a feeding link will only be generated if all of the traits are considered compatible. So in this case, only the middle consumer can feed on that resource. So once the model has done this for all of the interactions, it will return an interaction matrix showing all of the feasible feeding interactions. And it's from this point that we can now generate our meta community network. So we tend to visualize this network as a 3D web using iGraph. But you don't have to do it this way. You can plot it in any way you like. You can do a 2D network if you'd prefer. And one thing I really want to quickly point out is that in each of these networks, the different colors represent different trophic levels. And the way that this model assigns trophic levels is by the average distance of a particular taxa node to the primary node. And then from this point, we can now calculate some structural metrics and functional motifs, and these are all built into the model. And they're really commonly used network and node level measures, and they're really good at telling you a little bit about the network structure and what is actually happening. So there are a whole bunch of different metrics and motifs, but I'm just going to use 3 examples today. So I'm going to focus on firstly connectance. So connectance is the proportion of realized links out of all possible links which are present. So in the 2 examples here, the purple web on the left would have a higher connectance than the web on the right. And this really just gives you an idea of the density of links in a food web. The second metric I'm going to look at is generality. So this is essentially just the average number of resources a consumer node is feeding on. So the higher the level of generality, generally the higher generalism there is, because with generalism, there are more resources per node. So if you have a low measure of generality, then there's likely more specialists in your food web. And then finally, I'm looking at the functional motif of direct competition. So motifs look at the relationship between 3 connected nodes. So in this case, it is 2 consumer nodes feeding on 1 resource node. So these 2 consumers are directly competing for this 1 resource node. So before I go into my food webs, I'm just going to quickly talk about the data part of this workflow in regards to my research. So when the film was first designed, it was used for a data set which was predominantly benthic invertebrates. And because of this, they used the Bambac ecospace cube to determine traits. So the Bambac ecospace cube has tiering, feeding, and motility. So they use this in addition to body size to determine their traits and rules. And this is really, really great for invertebrates because this ecospace cube separates out the ecological guilds of these invertebrates really well. However, this wasn't the case for my data. So I got my data from the Palaeobiology database using the Pliocene and Pliocene as my pre and post extinction communities. And the majority of my data set was vertebrate genera. I did have some invertebrates, but I represented these as singular nodes. So I had 1 node to represent all microinvertebrates and 1 node to represent macroinvertebrates. I did also have 19 squid nodes because they play really important pelagic roles. But aside from this, these were the sole representation of invertebrates in my dataset. And this means that a lot of the traits that are used in the Bambac ecospace cube are redundant with my data. So if you take motility, for example, all of my data is fully motile, which renders the other categories redundant. So to make sure that I had adequate separation between all of my genera to infer the best feeding interactions that I could, I had to decide on different traits to use than were originally built into the model. So this was the list that I eventually ended up with. So I still have body size because body size is probably the most important trait when it comes to feeding interactions, and it basically is the foundation for optimal foraging theory. But in addition to that, I also had feeding habit. So feeding habit is pretty similar to the feeding category that we see here in Bambac's ecospace cube, but it's just a lot more specific. So where here they just have a category of predatory, I will have predatory, but then I will specify whether it's an invertebrate specialist, whether it can feed on fish, for example. And I'm able to do this because the extinction event I'm looking at is pretty recent. So a lot of the taxa in my data set are still alive today. So we can be pretty confident in knowing what these things ate during this period. So we can be a lot more specific with our traits. And then the final two traits that I used were depth distribution, so how deep an organism lived, and then also its feeding position in the water column. And these two essentially cover the spatial aspects. So because I had new traits, I also had to then develop new rules. But they followed the logic that the original dataset used, and very much underpinned by optimal foraging theory. So it has to be energetically beneficial for a consumer to feed on a resource, and they have to share the same spatial distribution. I'm going to go into this a little bit more later, but I just want to point out now that it's really important when designing rules that you have a lot of justification for what you're doing, because changing one rule can have a really large impact into the structure of the web, and you can get really significant changes by only changing minimal parts of these rules. So here are my meta-community food webs. So on the left, I have my pre-extinction communities, and on the right, my post-extinction communities. Before I move on to the metrics and motifs, there are just a few things I want to point out. So like I said before, the different colors represent different trophic levels, and the different nodes represent different taxa. And the way that the taxa are distributed in these webs is really similar to what we see in modern ecosystems, which is what we'd expect, because at least in terms of taxa composition, these Pliocene and Pliocene ecosystems are really similar to what we see today. So in trophic level 2, we have our herbivores and invertebrates. Trophic levels 3 and 4, we have our mesopredators, and then our very top predators are in trophic levels 5 and above. It is really interesting because in modern ecosystems, we normally only get 5 trophic levels in marine ecosystems. So here we have recovered an even higher level. And in this top trophic level 6, we have the megalodon, which is exactly what we'd expect as a giant apex predator. But we also have this sperm whale as well. And the sperm whale is still in trophic level 6 following the extinction. So the sperm whale often raises a few questions, because in modern ecosystems, we'd normally recover this species in trophic level 5. But it isn't unusual necessarily for it to be in this high trophic level, because it normally sits at the very top of trophic level 5. So it's not that different. And also, because these, because these are meta-community food webs, and they're showing all feasible feeding interactions, it's possible that the sperm whale here is feeding on more things than it would be in modern ecosystems, and that might be inflating its position a little bit. But at the same time, we can't say for certain that it didn't occupy such a high position during this period. So when we consider the metrics and motifs, when we're looking at how these food webs structurally changed before and after the extinction event, we can see that there was actually very few changes. So I'm going to bring up the 3 metrics and motifs that I spoke about before. So firstly, connectance. So we do see a slight increase. So the food webs in the Pleistocene are more highly densely connected than they were in the Pliocene. And similarly with generality, there is an increase in generality. So there are more generalists post-extinction than there were pre-extinction. And these two things can go kind of hand in hand, because when you get more generalists, you will have more feeding interactions. So therefore, you will inherently have an increase in connectance. When we look at direct competition as well, we actually see this decrease over the extinction event. So fewer predators are competing for the same resource. And we might expect this because a lot of the marine megafauna that went extinct in the Pliocene were predators. So there is some logic to that. However, I just want to point out that the magnitude of these changes is incredibly small. So, and this isn't just the case in just these 3 metrics, it's across every metric and motif we look at. There's very few significant changes in ecosystem structure and function over this extinction event. And this could imply that maybe these Pliocene marine food webs were pretty robust to these losses of marine megafauna, or maybe losing these nodes from the higher trophic levels doesn't have as many cascading impacts as we would expect. However, we wanted to make really sure this was actually the case. And there is one problem with meta-community food webs, and that's to do with connectance. So meta-community food webs, because they show all feasible feeding interactions, they're often more highly densely connected than you'd expect. So we wouldn't expect all of these feeding interactions to occur in real life. And because of that, we get a really high connectance value. So to put that into some perspective, in modern marine ecosystems, we'd normally expect a connectance between around 0.01 and 0.1. So this is obviously a lot higher than that. And when you do have really highly densely connected webs, you're at risk of potentially masking any structural signals that you might be seeing. So there is one way to address this, and that is to generate realized webs. So realized webs are essentially when you take your meta-community webs and you downsample them. So you randomly remove links to achieve a target link distribution. So this link distribution is based on a mixed exponential power law, which is what we see in modern marine ecosystems. So it's the same link distribution that we'd expect to see in a food web. And because we're randomly removing links, we're getting a very wide variety of structures. So we generated 1,000 iterations of these realized webs per time bin. So 1,000 for the Pliocene and 1,000 for the Pleistocene. This next step is pretty optional and very much dependent on your data set. So at this point, you can remove unrealistic topologies. And obviously, this is quite a hard thing to justify whether or not a topology is unrealistic, because we don't know what these food webs would have looked like millions of years ago. However, with my data set, it was pretty clear that we were getting some unrealistic topologies, and that's because there were some webs we were getting with more than one primary productivity node. So this happens when in the meta-community webs, there is a node which already isn't very well connected, and then it loses all of its feeding interactions. So, and that because of that, it gets moved to the very bottom of the web alongside the primary productivity node. So this is pretty reasonable to assume these are not realistic topologies. So these were easy to remove. And from this point, with the remaining webs, you can then calculate the structural metrics and functional motifs as you did before. But now, because we have such large sample sizes, because we have thousands of different webs for the Pliocene and Pleistocene, we can now calculate effect size using Cohen's d. So effect size doesn't measure the magnitude of change that we're seeing, but it's more showing the difference between the two groups. So it can kind of give us confidence in whether or not the change we're seeing is actually happening. And here's just a very, very small selection of some of my realized webs for the Pliocene on the left and the Pleistocene on the right. And immediately, the biggest difference between these webs and the meta-community webs are the fact that all of a sudden we have a lot more taxa in our trophic level 6 and even a few in trophic level 7. So this is kind of an inherent factor of when you're randomly moving links, because for some of the nodes, you're now creating much longer chains for them to reach the primary node. So you are increasing their trophic level. And because of this that we don't use these realized webs to infer specific positions of taxa, but we use it more for just understanding the metrics and motifs. So when we calculate the metrics and motifs again, we find exactly the same pattern as we did in the meta-community webs. So we see an increase in connectance, an increase in generality, and a decrease in direct competition. And as with the meta-community webs, these changes are very small. So although they are changing, the magnitude is very small. But now, because we have effect size, we can look into this a little bit deeper. So both connectance and generality have a large and a medium effect size. So this means that although the magnitude of the change is small, we can be pretty sure that it is happening. Whereas with direct competition, this has a small effect size with a Cohen's d of actually less than 0.1%. So this is very, very small. So all of a sudden, this change appears more negligible than it did in our meta-community webs. So it's really interesting to compare both of the results from the meta-community and the realized webs to get a better understanding of what is actually happening. And thankfully, in this case, the results did essentially align. So we can say that overall, there wasn't actually that much change in any of the metrics and motifs over the extinction. And that wasn't due to high connectance of the meta-community webs. We're still seeing this signal in the realized webs. So just to quickly finish off this portion of the talk. So I suppose there's 4 major takeaways from my research so far. So firstly, we can say that from these paleofood web reconstructions, there weren't any significant changes in ecosystem structural function across the end Pliocene megafaunal extinction event. And this could imply the ecosystems were robust, these losses of megafauna, even those in the really high trophic levels like the megalodon. And this potentially has positive implications for modern communities. However, we are under the assumption then that these modern communities are as robust as they were in the Pliocene, which obviously we don't know that. We do see some very, very minor changes across the extinction. So we do see an increase in link density and an increase in generalism. And this is a really common thing to happen following extinction events because extinction events normally target specialists, or at least they're more at risk of extinction than generalists. And then finally, I just want to point out that it is really valuable using both meta-community and realized webs when you're trying to understand changes, and they're both valuable in their different ways. Okay, so finally, I just want to talk a little bit about the impact of ecological traits and rules on food web topology. So this model, the FIM, has actually been in a published paper, and because of that, the R code for this is widely available and it's really well annotated, like it's quite self-explanatory. So I'm not going to go through that now. Instead, I'm going to talk about a Shiny app that we're currently developing. So this has been largely the work of Tanya Stradum, who's a postdoc at the University of Sheffield. And the eventual goal of the Shiny app will that it will become a tool which will allow you to upload your data set, so your taxonomic data set with your ecological traits, and then you'll be able to build the rule set in the app, and then you'll see the food web being generated as you change those rules. So it'll be really helpful to kind of see, like, as you change the rules, what impact this will have on topology. So this is publicly available. You feel free to go look at this GitHub. But at the moment, it is still very much a work in progress. So for now, I'm going to show this app, but I'm going to use my data and just look at one of the traits as opposed to all of them. And I'll be looking at body size. So my body size rules that I use for my food webs is that consumers can feed on resources in the same size category or up to 3 categories smaller. And here are my body size categories. So as you can see, they're already pretty broad. So we're already being really flexible with what taxa can feed on another. But this rule does differ from what we normally see in food web studies and what we saw in the original FIM design, in that we now have a lower limit on what taxa can feed on. So there's a limit to how small a resource a consumer can eat. And this doesn't affect all the size categories. So for example, if we have a large consumer, it can feed on itself, it can feed on something medium, small, or tiny. So essentially that has no limits. Whereas if we have something mega, it can feed on itself, something giant, large, or medium, but it can't feed on anything small or tiny. And we did this because this makes more sense energetically, because we were initially, when we did not have this lower limit, we were having things in the mega size category. So for example, the megalodon feeding on something that's like 30 centimeters big. So we had like the megalodon feeding on anchovies, which probably didn't happen. This is not a beneficial energetic trade-off. So in addition to this size rule, just one other thing to point out. So all of these size categories can feed on the primary node and the microinvertebrate and the macroinvertebrate node. So this basically allows for any taxa to be a herbivore or an invertebrate feeder, regardless of their size. So I'm going to quickly stop sharing this screen and open up the Shiny app. Hopefully everyone can see that. So it's a little bit confusing at the moment, so I'll just explain it. So on the left here, we have our consumer resource rules. So this is showing that for each of the size feeding classes, what they can feed on. So for example, within the mega feeding class, they can feed on all of these categories. And then it has this for each of the feeding classes. The current rules is essentially the same thing. It's just written out in list form. It just makes it a little bit easier to see. And then down here, under the static size rules, this is just my food web as it is. So this won't change as we alter these rules. And on the right, we have a dynamic food web. So this will change as we alter the rules on the left. So this is my data, and this is the same food web. Obviously, it looks a whole lot different than the food webs in my presentation because those were 3D networks, whereas these are 2D networks, and they don't have that same upside down pyramid shape. But it's exactly the same data, and it's the same color scheme as well. So we have our trophic level 2 in this light green, trophic level 3 dark green, 4 is the light blue, 5 is the dark blue, and then we have our trophic level 6 in the purple. So we actually changed the megalodon to a little triangle, just so you can kind of see where it moves when you change some of the rules, which can be quite interesting. So the eventual aim of this goal would be to have not just the size categories here, it'd be to have all of the different traits. So then you can go between them and alter the rules and then see how this food web changes as you do so. But for now, I'm just going to demonstrate how important some of these rules are. So what I want to point out is that very small changes can make really big, significant alterations to the food web. So if I was to do something, say, for example, if I was to take the tiny feeding crasses, currently tiny taxa can feed on other tiny taxa, and they can feed on the microinvertebrate, microinvertebrate, and primary node. However, a lot of my tiny taxa are small fish that feed on other small fish. So if I were to remove the tiny feeding category, so if I said tiny taxa can no longer feed on other taxa in the same size category, all of a sudden there's a whole bunch of taxa in my food web which don't have anything to feed on. So they now fall down to trophic level 1 alongside the primary productivity node, and now we get tons of these grey trophic level 1 nodes. And because of this, because we've essentially lost nearly a trophic level, all of our other taxa have dropped in trophic value. So we no longer have a trophic level 6, and our megalodon is now sitting in trophic 5 alongside the sperm whale, and they're the only taxa now in that trophic level. Obviously, that isn't necessarily a realistic thing that you would do. It doesn't make sense that you couldn't feed on something in your own size category. And this works the other way as well. So say I allowed my tiny taxa to now feed on small taxa. So they can now feed on resources in a size category larger than themselves. You then get the exact opposite happening. So whereas last time we essentially dropped the trophic level, we've now increased the trophic values of a lot of our taxa. So if we look at trophic level 6, there's our megalodon again, but now we have 4 taxa in this trophic level as opposed to the 2 that we originally had. And not only that, our trophic level 5 has expanded massively. The majority of the taxa are now in trophic level 5, and trophic level 4 is non-existent. So you can see that although it was only this one tiny change, we only let one size category feed on something larger than itself, and already it's had major alterations to the food web. But again, that isn't necessarily something that you would do. It doesn't make a lot of sense logically, especially if we're using optimal foraging theory. But something that I do want to point out is the importance of this lower limit to my body size rules. So as I said, a lot of food web studies won't have a lower limit, and that's probably very much dependent on the data set you're using, and it doesn't make sense here because of how large some of my taxa are. But if I were to, in the mega feeding category, so for taxa larger than 12 meters, if I were to allow them to feed on tiny taxa, so taxa within and below 1 meter, this again really changes the food web structure. So we've lost trophic level 6 again, and our megalodon is now sat in trophic level 5 down here. And this can have really big implications, not only for when you're looking at food web structure and function, but also if you're considering the specific positions of taxa. So another part of my research, which I didn't talk about here, is that I'm interested in the impact of extinctions on the trophic positions of top predators. And if I were to use this food web as opposed to my original food web, my food web down here, all of a sudden my position of the top predators have changed a lot. My 2 top predators, my megalodon and sperm whale, are no longer in trophic level 6. So if I'm then looking at this before and after the extinction event, this will have big implications for my conclusions. So one thing obviously that this app doesn't consider at the moment is the impact that these changes have on the metrics and motifs. So another thing that we want to add is we want another panel which will be able to calculate these as we go. So as we change the rules on the left and generate new food webs, it will also show us the values for the different metrics and motifs. So then we'll be able to also see how making one change to the rule can also impact these structural and functional metrics. But yeah, I'll be really interested for any feedback anyone has, if anyone wants to have a go at this. I'll share the GitHub link again at the end. But yeah, thank you so much for listening.